I am Dr. Lorna Lancaster, and I am going to be talking to you today about the road to freedom. And that's going to be a bit about vaccines and the formulation of vaccines and how vaccines work. And I will be talking a little bit about some of the COVID-19 vaccines that are now in use, and some of you may have been injected with. So I'm with the School of Pharmacy here at the University of Lincoln, and this is something that I have worked on previously, but for bacterial vaccines in the past. So, vaccines are the most cost-effective and easiest way to prevent disease. And a lot of the time when it comes to disease, it's much easier to deal with if we can prevent it rather than treating it once it's already happened. In most cases, vaccines are known for the active immunization. So that's where we inject something into the person to stimulate an immune response that will protect them against the infection that they're immunizing against. But there is also passive immunotherapy. We're not going to be talking about that today, but it can be used where antibodies can be used to treat the disease. But we are going to be talking today about active immunization. And it was first discovered by Edward Jenner, and he was around in 1749 to 1823, and he developed the first vaccine against smallpox. And way back when vaccines were first discovered, or before vaccines, or before Jenner discovered the smallpox vaccine, the smallpox killed a lot of people and was a big problem in terms of healthcare. But Jenner noticed that dairy maids did not get smallpox. And he worked out that what dairy maids did get was cowpox, which is similar to smallpox, but not as infectious and not as pathogenic. So they got a mild disease, but it wasn't a fatal disease compared to smallpox. So he took some of the pus from the cowpox pustules and rubbed it into a small cut on the arm of a child. And this happened in 1796. And then he exposed that child to smallpox, and the child did not get ill from it. And they say the first vaccination happened in 1796. However, there is some evidence that he may have used it on himself and his own children first. So possibly slightly earlier than that. So 50 years following Jenner's work, there was more and more vaccination happening within the world, and the number of deaths from smallpox fell from 23,000 to 5,000, and this was before it was made compulsory and widespread. And in 1853, so a good 50 years after or 60 years nearly after the first vaccination, the actual vaccination became compulsory to all infants within 4 months of birth. And since then, smallpox has been eradicated from the world. And in UK, vaccination was discontinued in 1971 because we no longer needed it, because we no longer had the endemic disease within our country. And then smallpox was eradicated worldwide in 1980, and we've not had a case since, with vaccination no longer required. And vaccines have been saving lives, not just for smallpox, but after the Second World War, a lot more vaccines were developed. And this shows here on the left-hand side, we have a graph showing the decrease in numbers of measles deaths and morbidity. So the larger one is morbidity, so those carrying measles or infected with measles, and that the smaller inset graph is from deaths. And going along, there was about sort of 600,000 to 800,000 cases in the UK. This is US, I should say. As once the vaccine was introduced, the first vaccine was introduced in 1963, and there's this very, very sudden drop-off to cases and in deaths at that time. And really since then, there's been very, very few cases and deaths, with the odd outbreak here and there. And in the other picture, I think it's a nice picture indicating some of the diseases that were problematic before vaccines and caused a number of deaths worldwide. But this is on the left-hand side, all the red vaccines. That's the number of cases of each of these diseases in the USA before vaccines were introduced. And then when they turn black, that kind of shows how many cases have happened since the vaccine's been introduced. And as you can see, most of them have turned pretty black, and there's a number of them with 0 cases anymore, and with a number of diseases being eradicated from the industrialized world. So how do vaccines work? So it involves stimulating the immune system. And the immune system has 2 forms, the innate system and the adaptive system. So the innate immunity is the first response and is involved in clearing infection. So it's made up of your skin, it's made up of the mucus in your membranes, but also there's a number of cells involved, and they do a lot of the clearing of the actual bacteria or virus from the environment. So they're known as phagocytes, and they basically eat these bacteria or viruses to remove them from the body. It's very generic and only recognizes something as foreign, but it will not remember them, so it will just eat anything that is foreign. The adaptive immune response is a learned response, so it's quite slow to get going because it has to be able to work out what the pathogen looks like. But once it's activated, it is highly specific and highly effective. So we have the in this the graph below. When you are first see an infection, so the primary antigen exposure, you actually have about 10 days before the adaptive response triggers and produces antibodies. And it starts off with some IgM and then some IgG. So a lot of the times the innate immune response has actually cleared the infection in this time, but your body has antibodies against this pathogen. So if you were to see it again, so the secondary antigen exposure, after only 2 or 3 days, you get this sudden surge of the IgG, and that's because the adaptive response is triggered first because you've got antibody response, and this leads to a sudden surge, and this means that you are probably not going to have any symptoms from that disease because it's cleared before the infection can take hold. So we're going to look at the adaptive immunity, and it's the body's ability to recognize and defend itself against distinct invaders and their products. So it's got 5 attributes, and it's very specific. It's highly inducible, so it's really good at turning on. Clonality means it's very diverse. It's unresponsive to self, so it means that it shouldn't attack the host's body. It should only attack foreign pathogens, and it has a memory. So the cells have a mechanism for remembering or recognizing the same pathogen if it was to re-attack the body. And this is what is harnessed when it comes to vaccines. So there are two main types of cells involved in the adaptive immune response. There's B lymphocytes, also known as B cells, and these are the cells that are involved in making antibodies. There's also T lymphocytes, or can be called T cells, and these are cell-presented antigens, so they interact with other cells to allow killing of infected cells. And both of these can be involved in the adaptive immune response for clearing an infection. And mostly we've been focusing on the B lymphocytes because antibodies are relatively straightforward to measure. But actually, in terms of COVID, we actually think that T cells are more involved in this type of infection. And there are a number of infections that do tend to have more involvement with T cells. So actually, a lot of research has been going into monitoring these more than compared to antibodies. But antibodies are extremely important in the adaptive immune response. And as I said, they are slightly easier to measure when it comes to vaccine efficacy. Both B cells and T cells are involved in recognizing antigens. Antigens are a part of the pathogen that the body can recognize. So they are molecules on the pathogen that are specific for that disease. And these tend to be something that the cell can bind onto and attack. Once antibodies are triggered, they have a number of different things that they can do to allow the body to fight disease. So antibodies can what's called do opsonization. So they bind onto the pathogen, as can be seen in figure A. So that's the antibodies, the Y-shaped structures, bound onto here a bacteria. The antibodies then bind onto a phagocyte, as we were talking about before. The phagocyte engulfs the bacteria and then destroys it. Can also be involved in agglutination, so that's stopping any bacteria or viruses from entering any cells or going to places to cause damage by binding to them and holding them in place. Neutralization, so this could be antibodies binding to toxins or certain outer membrane proteins. So, for example, in COVID, the spike of the virus. And the antibody binds on and stops the virus from binding onto the cell, or it can bind onto the toxin and stop it function. The last one is activation of complement and inflammation. So it triggers another mechanism of the immune system to attack the cell and is often used in bacterial or parasitic infections. T lymphocytes, well, there's two main types of cells that we're interested in when it comes to vaccines. So we have, first of all, a helper T lymphocyte or a helper T cell, and these activate phagocytes, so the cell-eating cells, and send out chemical signals to influence the immune response. So if they recognize an antigen, they tend to send out lots of signals using hormones called cytokines to go and recruit either the phagocytes or B cells for antibodies, and they also stimulate other cells to get active in getting rid of the infection. We also have a cytotoxic T lymphocyte, and these are involved in attaching to infected or damaged host cells to send signals to the cell to promote cell death so that the infection can be killed within that cell. So vaccines, how do vaccines activate the adaptive immune response? So they need to look similar to the pathogen, but not cause disease. So it's kind of like showing the body a picture. So if you have a villain, but nobody knows they're the villain, you need to tell everybody that they're not a good person to know. So you show everybody a picture of that villain. And that's kind of like what a vaccine is trying to do. It's showing the body a picture of something that might cause disease if they were to come across it, like as in the real villain. So we want to show it a picture that is very, very similar to the original pathogen, but not actually do the damage that the pathogen can do. So you need to expose the body to the antigen to provide the secondary response when it does meet the pathogen itself. So if we show our body that picture, so that antigen to begin with, you're stimulating the primary immune response, and which means that yes, there's a slow response to get the antibodies, but once the second, if there is a secondary antigen exposure, so somebody's exposed to the pathogen, they already have that memory response. So you get the IgG and IgM starting to come in as soon as the infection starts, so that they can clear the infection before disease can happen. And so there are a number of ways we can do this as scientists to make pathogens look like the original infection or the true pathogen, but without it causing disease. And the original method that was used was attenuation. So you can have attenuated vaccines, and these are modified live. And an example of what we have today that we vaccinate with is measles. We also have in other countries an attenuated live polio vaccine. And these vaccines contain actual measles or polio viruses. But what scientists have done is found a way that the viruses cause infection, but not in the cells they would normally target to cause disease. So in the case of polio, that's they infect the gastrointestinal cells, but not nerve cells. And this is very similar for measles. So if they're infecting the gastrointestinal cells, but not causing disease, the body is seeing still an actual infection, even if not at a level to cause disease. That means it stimulates an immune response very similar to the original pathogen. So it means you get a really strong, robust immune response that will respond to the measles pathogen when it comes and is fully functional. We also have inactivated vaccines, and these are wholesale. And the one that we use in this country today is for polio. And we basically take the polio virus and we treat it either with heat or with formaldehyde, which kills the polio so it cannot enter the cells and replicate. So our bodies are seeing the polio virus, just not in an infective manner. The advantage of this, there's less likely to be any kind of infection from the vaccine because they're not live. So it can be given to anybody, but it has to be given in larger doses and alongside other chemicals to try and stimulate an immune response, because their body is not seeing an infection as such. They're only seeing a foreign molecule. So we have to stimulate the immune response to be able to react to it. The other types of vaccines all are branched into a similar fashion of subunit vaccines. And these are, we're showing the body a small part of the vaccine, of the pathogen. So it could be a protein that's on the outside of the cell of the bacteria. It could be a spike protein on the outside of a virus, or it could be some of the polysaccharides that are on the outside of some bacterial cells. And we're only showing that one part of the bacteria or virus, which means that, again, there's no infectious substance going into the body, so anybody who's immunocompromised can have these types of injections. However, again, it's not as robust an immune response because it's not a true infecting agent. So you have to stimulate the immune response with other chemicals and adjuvants. We do have recombinant vaccines that can be used where you put the gene of a pathogen into a virus or a bacteria that doesn't cause disease. So it's a form of live attenuated, so the body will be observing a true infection as such, but without any disease occurring. And they'll be seeing one part of the pathogen that they can see. So that's one way that we're also using a live attenuated, but in a slightly different manner. When it comes to COVID vaccines, there's actually some new types of vaccines on the market now, and these have only come out really since the COVID vaccines have been established. So they've been in research for a long time, but they've not actually made it through approval so far, but they have now. This is where, instead of in the previous ones where we've been showing a direct protein from the pathogen or the whole pathogen in itself, we're actually providing a blueprint of the antigens. So this is instructions of how to make it. So it can be a DNA vaccine. There are actually none approved so far of this, and this is where the DNA itself is injected into cells, but there's a little bit of complication on how we get the DNA into the cells itself. The DNA can then be encoded and turned into the antigen of choice, and then the body will respond to that antigen. The other one, which is the Pfizer, BioNTech, and Moderna COVID-19 vaccines, as an RNA vaccine or mRNA vaccine. So this is where we put a little bit of RNA, which is again encoding for the antigen of choice. The cell makes it, and then the protein is available for our immune response, immune system to react to. The other one, AstraZeneca, the Oxford COVID-19 vaccine, is a virus vector. Some people would also describe this as a DNA vaccine, but they use a virus to allow the DNA to enter into the cell. So they put the DNA of the antigen of choice into an adenovirus. And the adenovirus, in this case, cannot cause disease. And the scientists have also changed it so that it can enter the cell, but cannot replicate. So they've removed the genes which allow replication of the virus within the cell. So it means that the adenovirus is very, very good at entering the cell. And once in the cell, the DNA is released to make the antigen. The antigen is made and then displayed on the cell, ready for the immune response. So this is kind of like a DNA vaccine, but with a delivery mechanism of a non-replicating virus vector. And they have used a replicating virus vector for the Ebola vaccine, which is in trials at the moment. And that's where, yes, the virus can replicate, but still not cause disease. So these vaccines work on the premise of the central molecular biology dogma. And this is where we have a gene, which is within the DNA. Once the DNA gene is selected, this is transcribed to RNA. The mRNA then is translated by the ribosome within the cell to make the protein. And so for the virus vector or DNA vaccines, they start with the gene in the DNA. But for the mRNA vaccines, they just miss out the transcriptions part of this pathway and just provide the RNA required to enter straight onto the ribosome to make the protein. And there are advantages and disadvantages of both. Okay, so this is just an image of the different types of vaccines that we can have. So we have the Pfizer-BioNTech vaccine, and we have the RNA in green in the center. So this is on the left-hand side. And we have a circle of blue circles or dots around the outside. And that blue circle of dots is to protect the mRNA. And this is the mRNA is encased in a micelle, and this is some fatty lipids that create a micelle, so like a bubble around the mRNA to protect it. So our bodies are particularly good at getting rid of RNA because we don't want it hanging around in our cells all the time. So we produce RNases, and RNases are everywhere. Okay, and if you're trying to work with it in the lab, they're really everywhere and they're impossible to get rid of. So RNA is extremely sensitive to the environment, so we wrap them in these fatty bubbles or micelles to protect it from the environment. These fatty bubbles also help the mRNA enter the cells. So these fatty bubbles can interact with the cell membrane of the host cell and allow the deposit of the mRNA into the cell to allow it to enter into a ribosome for the protein to be produced. Once in the cell, the mRNA only lasts for a few days once it's done lots and lots of protein production. And then, as I say, our cells don't want a lot of mRNA hanging around and over making lots of protein, so our body gets rid of it. In the AstraZeneca and University of Oxford vaccine, the green on the outside, that's the virus vector, and it's got spike proteins as well, which allow it to enter into the cell very easily, because that's how viruses like to enter the cell. And in the center, we have a DNA plasmid, which can enter into the cell once the virus has attached onto the host cell. The DNA is turned into mRNA and then turned into protein via the ribosome. So they look a bit different, but one advantage of the virus is that it actually can survive in sort of most environments, because that's what viruses are designed to do. They don't want to die off just from being at slightly warm temperatures. So the DNA is protected by the virus before it's being injected into the patient, and it gives it a more stable format for that injection to happen. So we only need to store it at 4 degrees, so the same as a fridge, whereas the Pfizer-BioNTech has to be stored at minus 70 degrees Celsius, so that we can protect the micelle and the mRNA from degradation at higher temperatures. So when we're formulating vaccines, as we're going to talk about, all vaccines do require some extra components. And as we were talking about, vaccines are made of biological products. So we need to consider the stability of the vaccine. A lot of our vaccines now are not live attenuated. So we need to think about boosting the immune response by the addition of adjuvants. And these stimulate the immune response by creating either a signaling pathway or inciting some inflammation to start the immune response from signaling to say that there's an infection present. There's usually some preservatives, again, to help with stability, and then some buffers and salts to keep the vaccine as happy as it can be. When formulating vaccines, you need to ensure that the antigen is the same structure as that within the pathogen. So any alteration to the antigen can result in the vaccine not being effective, because the antibodies recognize a 3D, very small structure, and any changes to those structures will cause the antibodies to recognize something very different to the actual pathogen that it will see if the infection was to come. You also need to think about storage. So the ideal storage really would be room temperature, but 4 degrees Celsius to allow it to be kept in the cold storage and delivered very easily. As we've seen with the Pfizer vaccine, there is some difficulty in delivering and maintaining that minus 70 degrees Celsius storage requirements. And that's in our country. Many countries don't have those facilities to be able to deliver that vaccine. And you also want a formulation that still allows the best efficacy for the vaccine and still remains effective. Okay. And as I've mentioned before, vaccines are biological medicines, so they're a lot more fragile to the environment compared to some of your small drug molecules. So we have to make sure that they are stored effectively and that they are formulated effectively to protect them from environmental changes. So we need formulations that allow stability, acceptable storage conditions, and acceptable delivery mechanisms. And some of the ways we can do this, we can have a liquid formulation that's stored at 4 degrees, and some will require that liquid formulation. We also have a mechanism where we can remove the fluid from a vaccine along with some caking agents and powders, and we do what's known as lyophilization. So the vaccine is dried to a powder, and then we have to reformulate it when it comes to actually delivering it. And depending on what type of vaccine, there might be quite a few procedures to follow to allow that lyophilization to occur. And we want the storage to be as easy as possible most of the time, but sometimes that's not possible. And we have sometimes storage at minus 70 when it's required. So what do pharmacists and pharmaceutical scientists do in the world of vaccines? So pharmaceutical scientists, so what I've been in is development of new vaccines, development of new delivery mechanisms. So it'd be so much nicer going for vaccination if it wasn't a stab in the arm. So scientists are looking at how we can take them maybe orally or with a little patch rather than a big needle stabbed in arm. Also looking at new formulations so that the vaccines are as stable as possible, preferably at room temperature, or even in higher temperatures to allow easy transportation of the vaccines, which can make them cheaper and easier for usage. And we also, because they're biological medicines, any slight change in the manufacturing of these medicines can result in a change to the vaccine or to the product. So every batch that's made has to be tested and made sure that they are still working compared to the original one that was licensed. So there are lots of scientists working on testing every batch and also testing all the batches to make sure they're working as they should do. So pharmacists kind of make the vaccines as best they can. And then pharmacists are there to help deliver them to the patients. So administer vaccines to the patients, advise patients on the use of vaccines, any side effects that they might have from that. They may be involved in the preparation of vaccine components, and then they're also involved in the research side of things and the sort of clinical trials often as well. So if you're interested in being a medicines expert, study BSc in pharmaceutical science to be the experts in design, formulation and manufacturing of medicines. Or if you want to be a pharmacist, it's to be the medicines expert within the multidisciplinary healthcare team, ensuring the right medicine gets to the right patient at the right time. And if you're interested, come and see us at Open Days. We'll be here. And if you just want to keep in touch, here's some Facebook and Twitter accounts for you to have a look at. And thank you for listening.